Now coming to the GI hormones. There are several GI hormones, but ideally at least we should remember the three of them. The most important hormones are the gastrin, cholecystokinin and secretin. All three we should remember. All three function from where they are secreted and their actions. So coming to the gastrin. Gastrin is specifically secreted by the G cells of the stomach. So this is the stomach diagram. As you can see here, we have drawn some G cell representation. From the G cells, it will be secreted. So what is the stimulus for it? The most important stimulus for it is the peptides. The peptides maximally secrete gastrin. And as the name gastrin indicates, what it is finally going to do, it is going to produce gastric acid. It is going to produce gastric acid. But right now it is not going to do that. Right now, first what it will do is it is going to stimulate two different cells. That is the parietal cells and the chief cells. It will stimulate them and ultimately whatever the secretion is coming from those cells will be secreted more. And whenever parietal cells is producing the HCl, what this H plus ions, will go and inhibit the gastrin. So gastrin is increasing their secretion, which is in turn is inhibiting the gastrin itself. So what type of feedback mechanism is this? This is a kind of negative feedback. It is negative feedback mechanism. So what are the functions of it? First thing is gastrin is stimulating two different cells. One is the parietal cell. Parietal cell, all of us know it produces HCl. Parietal cells is also called as oxyntic cells. It produces HCl. Along with HCl, it also produces one more substance is called as intrinsic factor. Intrinsic factor. This intrinsic factor is very, very essential for absorption of one vitamin. What vitamin it is? It is essential for the absorption of vitamin B12. So this intrinsic factor and vitamin B12, they will bind together and they finally get reabsorbed in the ileum. So if anyone is defective, then there will be a vitamin B12 deficiency. If the intrinsic factor is less, then also vitamin B12 deficiency will be there, or the nutritional factor vitamin B12, if it is less by itself, then also there will be a problem. So coming to the next cell, it is stimulating another cell called as chief cells. These chief cells are the one which is producing the substance called as pepsinogen. What is the function, other functions of this gastrin? It also increases the gastric mucosal growth as well as the gastric motility. So it is primarily involved in the stomach region. It is exclusive for the stomach region. It is produced from the stomach and it is activating all the cells in the stomach to produce these kinds of secretions. So coming to the diseases. What type of diseases can happen? Whenever there is a parietal cell antibody, parietal cells are the one which was producing HCl and intrinsic factor. Whenever there are some antibodies to it, what will happen? Both HCl secretion will be reduced and intrinsic factor will be reduced. This type of disease is particularly named as pernicious anemia. Pernicious anemia. So this pernicious anemia is happening because of which vitamin deficiency? It is happening because of the vitamin B12 deficiency. And less of HCl will increase the chances of infection because HCl is a strong protector against infections. So in this pernicious anemia, patient will have these two. Infection also will develop and they will develop vitamin B12 deficiency also. This vitamin B12 deficiency, be it whatever reason it is very, very essential for the maturation of RBC. So whenever there is less vitamin B12, the maturation of RBC will be affected. RBCs usually they will be bigger in size and as and when they mature, their size will reduce. So what will happen? Now this maturation is not happening, so the bigger cells will stay. That's why it is developing a typical type of anemia wherein we have the macrocytic cells and the anemia is called as megaloblastic anemia. So the pernicious anemia could be one of the reasons for megaloblastic anemia. And the person will have red beefy tongue. So this can be given in the history. All these things can be given in the history. And if it is given antibodies, then the pernicious anemia is the diagnosis. Then coming to the next disorder with gastrin, that is called as Zollinger-Ellison syndrome. What is Zollinger-Ellison syndrome? Zollinger-Ellison syndrome is a carcinoma of the gastrin cells. It is a form of gastrinoma. Here the gastrin producing cells or the G cells are into tumor and their secretions are increased. So what will happen? There will be obviously increase in gastrin, which is easily understandable because it is having a tumor cell. But what happens to the level of HCl? The HCl level will increase because gastrin is stimulating the peptic ulcer. Usually, this HCl will go and inhibit the gastrin. But can it happen here? It cannot happen here because the gastrin is directly produced by the carcinoma cells. Carcinoma cells. If the HCl is increased by any other reason, the gastrin levels should come down. But if it is increased due to a gastrinoma, both of them will be higher. The gastrin also will be high, the HCl also will be high. That is a point to be noted. Now coming to the second hormone, that is cholecystokinin. This cholecystokinin is produced from the eye cells of the duodenum. There is specific type of cell called as eye cells. And this cholecystokinin will act on the receptor called as cholecystokinin A. Cholecystokinin A. What is the most important stimulus? Whenever we think about cholecystokinin, all of us remember its one function, that is contraction of gallbladder. Contraction of gallbladder is involved in digestion of or emulsification of the fat. But the most important stimulus for CCK secretion is not fats, it's again the peptides, which is very, very important for its secretion. So peptides are the one which is the most important stimulus, even more than the fatty acid substance. This is one thing to note down. Next thing is, let's understand. So whenever CCK is produced from the cells, eye cells of the duodenum, what this CCK will first do is, it will go and stimulate the pancreas. First thing is, it is stimulating the pancreas. And it specifically stimulates the pancreatic acinar cells. There is acini part. When you talk about the pancreas, pancreas will have a ductular part and an acinar part. So there are two parts in pancreas. One is the acinar cells and other one is duct. And their functions is also different. Whenever there is stimulation of acinar cells, what will happen is there will be production of pancreatic enzymes. So this is for the pancreatic enzymes. Whenever duct is stimulated, what it is going to produce? Another important production from the pancreas, that is the alkaline secretion, that is bicarbonate. Bicarbonate will be produced. So first thing is, it is going to, CCK's most important function is to stimulate the acinar cells. So it is increasing the enzymatic activity. Next thing what it will do is, it will go to the gallbladder. This is our gallbladder and causes contraction of the gallbladder. It is not producing any kind of new bile. It is increasing the contraction of gallbladder, because of which the secretion or the availability of bile is going to increase. So second thing is contraction of gallbladder. Now it has produced the secretion also and contracted the gallbladder also. They have to reach the duodenum. There is one sphincter which is controlling this action. This sphincter is called as sphincter of Oddi. This sphincter is called as sphincter of Oddi. So it has to be relaxed so that the secretions can reach the stomach, the duodenum. It will cause the contraction of the pyloric sphincter. Why it should contract the pyloric sphincter? Because we don't want the secretions of the duodenum to go back into the stomach. To prevent this, it is contracting the pyloric sphincter. So this is again an important function. Then it increases the intestinal motility. Like gastrin was increasing the growth of GI mucosa, it will go help the growth of pancreas. And one more thing is, it augments secretin. Secretin and CCK, both of them are like friends. What they do is, they augment each other. They augment, one person will help the other person. So this will augment function of secretin and secretin will augment the function of CCK. So these are all the functions of CCK. Just remember this diagram. So the pancreatic enzymes, what they do, whenever they come into action, they will digest it and form more and more peptides. Again, peptides will increase the CCK production. So what type of feedback is this? This is a kind of positive feedback mechanism, because everybody is helping and increasing the next persons. So it is a kind of positive feedback mechanism. There is two terms. One is called as cholegog and cholerectic. This is also commonly asked. What is cholegog? Cholegog is any substance which increases the contraction of the gallbladder. Now we saw one substance which is increasing the contraction of gallbladder, that is CCK. Cholegog is a substance which is increasing the contraction of gallbladder. What is the most potent cholegog? Answer is again CCK. Then there is something called as cholerectic. What is cholerectic? If any substances is increasing the production of bile, cholegogs, they contract the gallbladder, they don't produce the bile. Cholerectic, substances which increase the bile production itself. And the question arises, which is the most potent Colorectic, it is the bile salt itself. The bile salt is the best stimulator for itself, and it is producing lots and lots of lots and lots of bile. So this is all about the CCK. Now coming to the third important hormone, that is secretin. Secretin, we have very few function. Secretin is produced from the S cells of the intestine, and this is the first hormone to be identified. So it is also produced in the intestine, and it also goes to the pancreas. But in the pancreas, it stimulates the duct part. It is stimulating the ductular of the pancreas, ductular system of the pancreas. Whenever the ductular system is stimulated, what is going to happen? More than the enzyme, the bicarbonates will be produced a lot. There is huge production of bicarbonate. So the bicarbonate levels are being increased a lot. And what is its other function? It will help its friend, the CCK. These are all its function. Now coming to another hormone which is produced in the GAT, but recently its name is changed, so that's why we are trying to mention it here. That is called as GIP. This GIP, all of us knew this GIP was a gastric inhibitory peptide, but right now they have changed the name itself. Why they have changed the name? Because in physiological doses, the function of GIP is not gastric inhibition. In physiological doses, its function is to produce insulin. Its function is to produce insulin. That's why it is now called as the glucose induced insulinotropic factor. It is again GIP only, glucose induced peptide or glucose induced insulinotropic peptide. The GIP, the short form is same, but its function is different, so they named it like glucose induced insulinotropic factor. It is inhibiting the gastrin secretion, but only under supraphysiological dose. It can inhibit the gastrin secretions, that is gastric acid secretions. So that's why they have changed the name to this another new GIP, because of its role to produce an insulin.